Biology chapter 19, theory video 13, gene therapy. Gene technology has rapidly advanced during the past few years, past actually 20, 30 years. The scientists figured out that if you can identify the genes responsible for genetic disorders, and then if you can uh, remove these genes, or if you can insert or correct these genes, it would they would be able to cure these genetic diseases uh, by inserting the normal alleles and also the correct gene. So this is the process of gene therapy. But this is not as easy as the theory would suggest. So uh, the genetic correction is not going to be easy, because there are problems that would originate getting the normal alleles of the genes into the person's cells, and then also getting them to work when they get there. So because of these are actually complications. In 1990, they carried out the first successful gene therapy for SCID, ADA-CID disease. Her immune system did not work. This child on whom the gene therapy was carried out, immune system did not work, and they have to actually live in bio bubbles, plastic bubbles, to protect them from infections, because even a single small infection can kill them. What they did was, uh, they removed some of the T lymphocytes from the child and added the normal alleles of the ADA gene using a virus as a vector. And then they replaced it, and the child was able to live a normal life for some months. But this is not a permanent cure. It has to be done every 3 to 5 months to make the immune system functioning properly. Then in 1992, gene therapy using stem cells harvested from bone marrow became successful. But there were problems initially. In 2004, children who had obtained this gene therapy uh, for SCID, they developed leukemia because of the retrovirus that they used as a vector. So the retrovirus will insert their genes into the host genome. So instead of that, now they use adeno-associated virus, AAV, which would not insert its genes uh, into the host cell. So as a result, it will not be passed on to the daughter cells when the cell would divide, and this is really successful. And from then when it was in 1990s, now they don't need regular transfusion of T lymphocytes, so it is kind of permanent, or at least long lasting. Then another condition that has been treated with gene therapy is Leber congenital amaurosis. This is actually an inherited blindness, Leber congenital amaurosis, LCA. So it actually causes the retina to die off from an early age. This can be reversed using gene therapy. So what they do is, there is an recessive allele that causes the vision to be impaired when exposed to light. So what they do is they can reverse it by inserting the correct allele, or the repaired allele, into the cells who are going to be homozygous recessive for this gene. And also, because this is a very small organ, it is actually a very good target organ for gene therapy, because it's really small, and genes can be delivered precisely into the retina. And also there are only little activity of immune cells inside this eye, so there is a low risk of harmful immune response to the vectors as well. So this diagram will show how it is done. This is actually the procedure for carrying out gene therapy for SCID. First, what you do is uh, you take the DNA with correct base sequence for ADA prepared. Then using the adeno-associated virus, ADA, you insert the normal allele of ADA into the virus, and it will be delivered into the uh, uh, cell. That child with ADA CID will give the stem cells from the bone marrow. It would be removed, and virus will insert the normal allele here. So what needs to happen is when this after this step has taken place, this needs to happen. So the stem cells would be removed from the bone marrow, and the virus will insert the normal allele of ADA onto the stem cell. Remember, the virus will not transfer its genome into the cell, only the required gene. So then the stem cells with normal allele of the ADA are injected into the blood, and they will divide and differentiate into T lymphocytes that have functioning ADA. I hope you understood. If you have any questions, please ask. Let's write down a note on this. Gene therapy is the treatment of genetic disorders by inserting genetically corrected cells into the body or introducing functioning genes directly into affected cells. This has the ability of curing or reducing the effect of genetically transmitted diseases. For example, the disease ADA-SID, which results in an unfunctional immune system, can be corrected by inserting the normal allele of the ADA gene to the T lymphocytes using adeno-associated virus AAV as a vector and injecting these corrected lymphocytes into the bloodstream of the patient. These T lymphocytes will divide and provide the necessary AAD. Another disease that can be cured by gene therapy is Leber congenital amaurosis. This is an inherited form of blindness causing the cells in the retina to die off from an early age. This can be reversed using gene therapy. By inserting the dominant allele of the gene RPE65, which codes for a protein that regenerates visual pigment in rod and cone cells in the retina after they are exposed to light. This is comparatively an easy process of gene therapy because eye is a small and easy target, resulting in genes being able to be precisely transferred and being able to monitor the progress of the treatment easily, and also because of the low immune activity, there wouldn't be a harmful immune response. I hope you understood. If you understand, please do question number 16, 17, and 18. And if you have any difficulties, please ask me. But before that, I would suggest that you draw figure 19.27 also to give a clearer idea for you. After that, please do the questions.